Okay, so I want to welcome everybody back. If you were with us with the first hour, the total gut restoration, now we are going to start the second hour of the BiomeFX stool kit that Microbiome Labs has launched. You'll see this terminology, shotgun metagenomic sequencing. This is just a term that's referring to the fact it's whole genomic sequencing. This is a quote by Kieran. There is a real need in the functional medicine industry for a stool test that is accurate, reliable, focused on key functions, useful for therapeutic recommendations, and easy to understand. So Kieran made this statement because he was in search of finding a test out there and he could not find it. You could do any of the tests except from BiomeFX on a Monday, Tuesday, and Wednesday, get your test results back and have 3 different tests. There was abominable reproducibility. So that, is the reason you won't find any of those tests being used in mainstream universities, institutions, or agencies. But you will find BiomeFX, i.e. Cosmos ID, in those institutions, universities, and agencies. So the objectives here, why we chose the particular stool test we did, what sets it apart from other tests on the market, and review the sample report. Okay. So effectiveness. Of course, we wanted to create a more effective tool. Functionality. Offer a bigger picture of looking at the gut that offers actionable items, lifestyle nutrition supplements tailored to one specific microbiome, fill a need. What is currently offered lacks specificity, accuracy, and actionable steps. So today's stool test challenges in the market are specificity and sensitivity. The data and analytics are not reliable. The interpretation doesn't really translate well to individuals, and the lack of functional information. What do you do with the data that you have? So Kieran searched, took him about 5 years, and found Cosmos ID. And it is the highest degree of accuracy on the market, many competencies in place to prevent biases, reference ranges based on 426 healthy subjects, and how they got these. Again, remember, they were the ones that did the testing for the Global Microbiome Project and the American Gut Project. And they were able to amass the world's largest database with 160,000 microbe genomes. They have functional insights on how the microbe and host interact, third-party testing, most accurate, precise lab. They do the whole CLIA and GLP certified. And both Ken and I have worked in labs, and those are not easy. Let's just tell you that. Now, this shows you Cosmos ID is involved in the pharma industry, clinical, nutrition, agricultural, consumer, applied testing, and this is showing you the name brands. And believe me, these people's names are on the line. They're not going to use an inaccurate test. This is important to understand. You think back, to your BACT, microbiology, immunology classes, on the taxonomy of organisms from kingdom, phylum, class, order, family, genus, species, strain. Okay, so keep that in mind. That is how you catalog species. For humans, it's here down to Homo sapiens. For a bacteria, it comes down to a strain number. Here, you might put a social security number if you want a human strain, right? Okay. Current testing challenges. 16S amplicon methodology. So this is the most popular current methodology in the market. It looks at the genus level instead of the species level. Only covers 1 to 2 hypervariable regions on a gene. A given hypervariable region does not capture all the bacterial lineage equally well. So let's explore what that means. This is an amplicon. So this is the region that you're targeting, and you will splice a section, and then through PCR technology, you will amplify it. All right. Now, there are problems with that. It's not the whole genome. It's just a piece, right? So you can take a piece and you really can't identify, even though it takes several pieces, you're still ultimately guessing at the identification. So there's high false positive rates. Artificially amplifies genes cannot identify viruses, fungi, and protists, cannot identify the functional inferences, cannot identify antibiotic resistance. So this is what whole genome sequencing will do. It will give you the identity. End-to-end sequencing of DNA, able to analyze at the species and strain level, identifies bacteria, virus, fungi, and protists. Most accurate testing on the market, 99% specificity, 95% sensitivity. So we partnered with them. Our test is called BiomeFX. And this, I think we mentioned. Now, this is kind of illustrating the coring technique, which is a device that is going to give you a more representative sample. So, you know, some technologies will just kind of, you know, scrape the top of a stool. That, you know, unless you get into the inside of the stool, you don't have any representation higher up on the GI tract, right? So that's a huge improvement. All right, now I remind you of this ranking. One of the things when you get your sample test back, the last page actually gives you your 30,000 foot view, and that's the phylum, right? So that's what this is. You're looking at the phylum of what a healthy population looks like, and then this is the sample that you turned in. So the first thing, and you hear a lot about the ratio between Bacteroides and Firmicutes. I think in the first, I've been with Microbiome Labs 4 years. The first year we had the science that, you know, if you have more Firmicutes than Bacteroides, you're more likely to have weight problems, right? And who's not interested in that? So this kind of shows you right off the bat, you know, in the red is the Firmicutes. You want to have more Bacteroides than Firmicutes. A healthy sample will have more Bacteroides than Firmicutes. The sample that, you know, you turned in has too many Firmicutes. So that's the first thing you can see right off the bat. Wow, that's why I have, you know, this extra tire. My gut bugs aren't helping me. All right, so that's the first thing you can look at. Can I get an idea for what community you're dealing with? The second here, well, that's the same, phylum. Phylum is the biggest grouping bacteria, good visual to reference all of the data on the test. Then you've got the family view. Same thing, this is what you should see, a lot of, you know, different colors here. Here you can see there's not as many colors. So you, just as an impression, you can say, you know, we've got some diversity issues going on here. So this is the sample report. The first thing it gives you is an index, and this index is based off of these first three, which is the alpha diversity, beta diversity, and resistome. So, you know, it's giving you, do you have a good combination? Do you have the right combination for where you live? And is it going to protect you? So that's what you're going to see first off. So this person's doing pretty good. And that's just what I said here. Anything above 30 is excellent. So what is this alpha and beta diversity part? You know, and again, there's some scientific papers I had on a link in Constant Contact, and what you'll see are all of these informatics. What you'll see, our test is, you could go into a scientific lab, a research lab, and you'll see they're using these type of tests, these type of statistics, these type of graphic representations, because this is the cutting edge stuff, right? So the definition, Alpha diversity is talking about species richness, the average diversity of a species in your gut. A low alpha diversity suggests that you've had antibiotics, or you've wiped out some species, and what will also do that is stress and toxins and damage to your microbiome. High alpha diversity is paramount to health. This is why we're always pushing, you have to eat a variety of food to keep a variety of organisms, improve the resilience and resistance of your microbiome. It's tied to longevity and chronic conditions, linked to better short-chain fatty acid production, and overall increases metabolic production. Beta diversity, average overall composition or variation of species when compared to a healthy population. High beta diversity, we're looking at this red dot. You want it to be right in the middle, and this is right smack in the middle of the crosshairs here, so that's perfect. Compositional similarity, successful coexisting within the environment. I mean, if it was way out here in one of these quadrants, then you would wonder, was this person from a foreign country, because, their organisms aren't similar to the organisms in the geographic area where they live. Okay. The resistome. Okay, so this is not what you would think antibiotic resistant. It's all organisms produce antibiotics. They all have boundaries among themselves, right? So this basically shows whether the ecology is off. It's saying, do your gut bugs get along with each other? That's the bottom line, right? It indicates uniformity, stability, equilibrium within the microbiome. Because if everybody's happy, they're going to protect you better, right? Against COVID or parasites or whatever is challenging you in your environment. Pathogen control index. All right, so this index is looking at how capable your microbiome is at controlling pathogens, indicates strength and stability. So the higher the score, the more capable your microbiome is able to control a pathogen. So just because you got some E. coli there, your microbiome's handling it fine. Don't go in there with an atom bomb. It's not necessary. Okay. All right, these are now the ratios. We already talked about the Firmicutes versus Bacteroides, and you want more Bacteroides than Firmicutes, so that's one of the ratios you look at. Also, Proteobacteria to Actinobacteria is an important ratio. Most of the pathogenic organisms are in the proteo, like Shigella, E. coli, Klebsiella, and your good guys are in the actino, the bifido. So, there's a certain balance you want to keep between these. You don't want to eradicate these guys. It's not that you're not supposed to have zero. They're just supposed to be in balance, under control. Otherwise it's racial profiling. It's not right. This is Proventella versus Bacteroides. Another important ratio. And this shows the gases, and it's not enough to know, are you producing ammonia? You also need to know how your body's dealing with it. Are you clearing it? Ammonia is very important. It destroys your liver. So it has to be kept under control. We're also going to look at sulfate and methanogens. We're going to go into the estrobolome in great detail in the next lecture. Keystone strains. We've emphasized that over and over. Very, very important. These are the guys that protect you from inflammation and help your short-chain fatty acid production and keep your mucin levels intact, and yada, yada. So specifically, the Akkermansia is the primary mucus degrader, promotes mucosal health and mucus production, low levels associated with obesity, metabolic dysfunction, intestinal permeability, and can be stimulated by fasting. This is, your fat burner. So you want more of this guy, right? Faecalibacterium pranitsii, this is important species in the Clostridia class. It's a major butyrate producer. It promotes anti-inflammation and mucosal homeostasis, can reduce levels associated with wide range of chronic inflammation and autoimmune disease. Okay, and then here's a link. And I'm going to talk about this in the next lecture, all sorts of training videos done by Kieran himself. This is a little kind of marketing flyer here. We talked about the coring, the whole genomic sequencing. Okay, and this is just a slide on the ordering. So, you know, today we're going to get a discount code, and it's going to take, for anybody you're registering for the test, it's going to take 10% off of this. So that's great. You're going to save about $30 for every test that you submit, you know, because you were participating. Also, we have consults. We've got Lacey, who is just wonderful, and then Dr. Maltz, who specializes in helping you go over the test result before you talk to your client, so that while you're learning this, so that you're competent and prepared. Okay, so when you order the kits first, and you order them when you order products, so that you're not charged any extra shipping, and then you're only charged when you register the kit online. So you're going to get some extra time because some of you may not have kits on hand. So get those ordered, and then you know, I'll help you get the code so you can register and get your discount. All right, so that's the overview, and now we're going to go deeper. All right.
All
right. Shut down some of these. If you weren't with us in our previous lecture, know that all these links are in the signature block of all my emails. So you've got all of these YouTube videos which cover all of this information, as well as the protocol that's on all my emails. So know that those are there. Okay, so now we're starting next lecture. And text us if you have questions on this second hour. All right. So the first introduction. So we went over the BiomeFX, who, what, where, when, why, and how to order. We didn't really go over my personal story, but just know that, or really date myself. I used Great Smokies back in the day before they became Genova. I used a lot of diagnostic, but you know, it's nothing like we have in BiomeFX. And yeah, we already mentioned that all the credible universities, research facilities, and agencies use Cosmos ID. So the objectives of this lecture is to really go over some of the key interpretive concepts. You have to find out your way of describing what you're looking at. And so I want to just give you some terminology and some options. I also want you to see the power of the potential of this test. We've rolled out phase 1. There's going to be phase 2 the end of July and phase 3 at the end of October. They didn't want to wait and to another year to get it all together, because just what we had in phase 1 was so incredibly important, and blew away, as far as accuracy, bacterial identification, than any of the other tests. But what you're going to see as these things are rolled out, I think will blow your mind. All right, and then we'll do some case studies to connect the dots, give you some pearls, hopefully get the light bulbs to go off. And now we talked about the videos. These are the videos. There's a link for each one of these. He goes into depth, again, why we created the stool test, why we partnered with Cosmos, who Rita Caldwell is. And this woman is amazing, has over 800 publications, currently working on the committee, helping with COVID. I mean, She will be our keynote speaker in Scottsdale, Arizona, where we're going to do the Microbiome Labs keynote conference. So all of this information is groundwork to better prepare you for when all the gurus converge in Arizona in November 13th. So that's really where you can nerd out. This is just a sampler. The video here will go over the BiomeFX, the limitations, the methodology, how long before patient sees changes, sample report, and it's broken down, alpha, beta, resistome, patho, dysbiosis, biologically active bacteria, and keystones. Yes, everything that we just talked about, but you're hearing it from Kieran. Every time you hear these, you hear something different. You get more information. I recommend you hear it from Kieran, from me, from Acacia, from Lacey, and then get it in your own words. And then call me, and we'll go over that until you know it forwards and backwards. This is a link so it's handy so that you can schedule your consult. As soon as you get back your first few tests, have Lacey or Dr. Moltz go over it with you until you're real competent. And then you've got a printed interpretation guide, and then this is those video trainings again. All right, so let's start again with the whole diversity topic. Whether you describe it in terms of richness contrasted with composition, or quantity contrasted with quality, or interstate contrasted with intrastate, it's all about what you're comfortable with. I think this is a great question. Your alpha diversity is telling you how many species inhabit your gut. Your beta diversity is telling you, do the right species inhabit your gut? So whatever resonates with you, how you describe this to your patients, putting it in your own words is, where you want to go. High alpha, protective, and low beta, not protective in your environment, means you have many different species, but they are different species from the US population. And then you have to sit down and talk to this person and ask them why. Have they traveled abroad extensively? Have they lived abroad? Do they eat radically different than everybody else in America? Which hopefully, they're not eating the standard American diet. But anyway. The only way you can have accurate reporting is to have a representative global library of samples. Now, we mentioned this, that only Cosmos ID achieves this because it is the lab that was used for the Microbiome Project and the American Gut Project. The gut microbial populations adapt to the environment. Ratios change, levels of seeded keystones. Now, you don't replant, you don't reseed, you change ratios, okay, are influenced. Pathogens are encouraged or discouraged by terrain. Now, these EGAN vectors, when we talked about the crosshairs, those are, you can Google those. Those are statistical latitude and longitude lines for each species. So it's pretty fascinating in the whole research world of this, that that's, how they refer to species on where they fall on, that diagram. The resistome, again, this is how resilient is your microbiome. And the reason it has to do with economics is because this is a community. They produce things, they produce metabolites for each other, they help their neighbor, or, if they produce bad metabolites, or, they can hurt their neighbor. So if you've got a community that helps each other, then that's going to help you, right? So are they thriving, or are they being suppressed? Are they getting along with each other? Healthy competition is good. If there's not enough competition, then the ecology's off. Someone is trying to be a dictator instead of a democracy. So healthy boundaries are good. This shows stability, uniformity of a society, and are dependent on individual freedoms of expression. It's almost like I'm talking about our Constitution and the Bill of Rights, but I'm talking about the microbiome, and the same type of rules apply. You know, bacterial equilibrium is their expression of their Bill of Rights for equality. Balance or homeostasis. Imbalance will be reflected in the alpha and beta diversity. A rich species mix, well adapted to the environment, will have high resistance, a good resistome. Okay, so the pathobiome, this is the presence or occurrence, not always an indication of dysfunction. Abundance, not always an indication of dysfunction without taking into context the healthy bacterial ratios. Effectivity or strength. How effective or strong is your microbiome at dealing with the pathogens? And this is represented in the control index. Higher, the better. It indicates that the pathogens are being dealt with well or being controlled by the commensal organisms. As an example, if a policeman had never come into contact with any perpetrator or any criminal in his life, how effective would he be? The criminals in our microbiome are the pathogens. They keep our immune system sharp. If we have good bacterial ratios and populations, they don't harm us. So that's just an example. Higher the number of pathogens that are being dealt with well is a sign of a healthy microbiome. There are no, and if you look at the other laboratories and they tell you, do this, you need to eradicate this, or, go after this with, killing agent protocols, there's no studies to support that. That targeting specific pathogens are helpful, especially not long-term. Dysbiosis ratios, your Firmicutes versus Bacteroides, high obesity. So in this ratio, if it's high, meaning more Firmicutes, then that is going to be a predication to obesity. If it's low, then you're better at reducing inflammation. In the proteobacteria, if it's high, you have a poor metabolism. If it's low, you have a healthier metabolism. In the Prevotella ratio to Bacteroides, if it's high, you have a low body mass index and reduced inflammation and representative of a carbohydrate fiber consumption. If it's low, you have more obesity and representative of a high protein and fat consumption. So just some little hints when you're reading that report. The biologically active bacteria, again, we go into the different gases and why they're important. The high ammonia, these are bacteria converting more protein into ammonia, which stresses the liver. It increases the pH in your gut, and you're more susceptible to pathogen, fungal overgrowth, and dysbiosis to leaky gut. And the sulfates, high sulfate producing bacteria means foods that tend to be high in sulfates, don't clear it well, it causes you more problems. You're going to convert more sulfates from these foods into hydrogen sulfide, nasty smelling gas, which then becomes toxic and inflammatory to your bowels. The methane, people who tend to have low sulfate producing bacteria tend to have higher methanogens because they produce less hydrogen sulfide. They're producing more methane, i.e. nasty smelling gas, from the metabolization of food. The methanogens produce higher levels of methane. These people have gas responses from their food rather than inflammatory response. Now, the estrobiome. There are about 60 types of bacteria associated with high beta-glucuronidase activity. So this enzyme, beta-glucuronidase, conjugates estrogen into its active form, i.e. bad. Increased activity is associated with high fat, high protein, and increased circulating levels of estrogen. Alternatively, high fiber diets are associated with decreased beta-glucuronidase activity. The reason this is important is because if you're unconjugating estrogen and you're recycling it, you're going to be more estrogen dominant. And estrogen dominance, for those people that deal with hormones, is one of the primary contributors to PCOS, to PMS symptoms, to all hormone symptoms come down to how much estrogen you have. It's one of the high symptom producing hormones. It is my belief that you must know the gut environment before you introduce any hormones into the body. Because if you cannot properly clear these estrogens, you're going to cause harm to your client, patient, guest. Okay, functional keystone strains. We talked about, the top 3, but, let's go a little deeper. The Akkermansia muscipula, again, it helps with metabolism and short-chain fatty acid production and mucin layer. The Ruminococcus bromide is a cellulose degrader, helps with skin health. Faecalibacteria panitzii helps with inflammation and short-chain fatty acid production. Ruminococcus flavidosus is a cellulose degrader. Roseburia intestinalis is a beta-mannan degrader and a butyrate producer. Eubacterium rectili is a butyrate producer. Bifidobacterium longum, acetate producer. Lactobacillus species is a lactate producer. Again, helps with pH balance of the gut. Now, we're looking at some previews of what is going to be introduced in phase 2. Saccharolytic fermentation. So high levels of saccharolytic fermentation are associated with healthy metabolism and reduced intestinal pH, which supports a healthy gut environment. Sac fermentation levels may be low as a result of high protein diet, low fiber diet, or insufficient keystone species. And the relevant bacteria you'll look at to determine this are the Ruminococcus bromide, the Lactobacillus, and Bifido species. So what essentially, we're in a society where there is such a spectrum of different diets a person can be on, but there's very little studies to show what diet is the healthiest for that individual. Tests like this will be able to help guide you as to what diet your client is on. Is their vegan diet the best for them? Is their 100% carnivore diet the best for them? Or should be somewhere in between? So this will give you some scientific data to really base your recommendations on. There will be, specific levels for butyrate production, propionate production, and acetate production. And if we go into, the importance of each of those different short-chain fatty acids, you'll see that they're fuel for enterocytes, scavenge ammonia, regulate the immune system, reduce oxidative stress, requires an acidic environment in the gut, and then the relevant bacteria associated with those are listed. The propionate short-chain fatty acid encourages regulatory T cell differentiation in the gut, promotes gluconeogenesis in the liver, supports insulin sensitivity, improves gut hormone production, works with butyrate to support healthy inflammation responses by inhibiting histone deacetylase in the macrophages and dendritic cells. Think about all the questions you have about histamine, okay? This is gonna help you really get to the root cause of that. Acetate production by fermentation of prebiotic fibers like insulin, GOSR, unabsorbed peptides, and fats. It's supposed to be fats. This is just a little recap that, these are the primary signalers among cells and organ systems. So, they provide energy for your gut epithelial cells, help with repair. Acetate and propionate are involved in the lipogenesis and gluconeogenesis. Low short-chain fatty acids, you're going to see, microbiota alter, colonic energy harvest, host gene expression, and increase inflammation. There will be information about TMAO. This greatly impacts the, cardiovascular it's all metabolization that's happening in the gut bacteria. So you can see if someone is at risk. Bile salt production, we already talked pretty extensively about 2.5 detoxification and how important the bile is. And if you're unconjugating it, how you're recycling these toxins and increasing the risk of SIBO, diarrhea, constipation, and bloating. Phase 3, we'll go more into things like erythrolins that have to do with longevity, aging, recycling damaged mitochondria, and regenerating new mitochondria. We're also going to be giving indications for histamine and indole production. So that's all cool stuff. But here's the cool stuff. All right. So this is just an overview. We've touched on this in different lectures. We know that our gut bugs produce hormones and neurotransmitters that alter behavior. They influence mating through pheromones. I'm going to repeat that, the very end, that is it your gut bugs that picked out your husband or wife, or was it you, metabolism and appetite, affects your immunity, alters androgen and estrogen and prolactin levels for gender. Okay, so who's really in charge here? So this is a great list here, and I gave you the reference for this publication where you can see what species of bacteria promote what neurotransmitter. Okay, so inductively, what we'll be able to do is tell you your propensity to manufacture serotonin, dopamine, all the neurotransmitters, GABA, based on what bacteria are identified by the BiomeFX. When you get your mind around that is wild, okay? So you'll be able to see what organisms are affecting the hormones and neurotransmitter that are, relating to a certain behavior, depression, anxiety, schizophrenia, bipolar, post-mating. I don't, oh, that's these are the pheromones that are involved in that. Sex hormones and reproduction, if you're dealing with infertility, let's look at the gut bugs, right? Growth issues, let's look at the gut bugs. So this is cutting edge right here. And this is really exciting too. You know the relationship between more females have autoimmune disease, correct? We know that. So this is an experiment that showed this model describing the pathway leading to male protection. So what they did is they took a male microbiome and put it into a female, right? So it enriched the species of the SPF, the segmented fibrous bacteria, and E. coli and Shigella-like. And they found that the testosterone enriches the male microbiome with specific bacteria, including those species I mentioned. Together, they are proposed to activate the immune system and lead to gender-specific protection from autoimmune disease. So transplantation of the male microbiome into the female elevates testosterone levels and leads to protection of autoimmune disease. Is that not wild? Okay, so that just shows you, now this paper I've had, for over 4 years, but now that we have a test that can show you the bacteria that we can directly relate to the hormones and the neurotransmitters, we can really use this information. So this is worth every penny you paid. So, okay, now let's do some case studies. So we've got a 35-year-old woman, Hashimoto's disease, autoimmune antibodies are around 300, many food and environmental sensitivities. She received antibiotics, confirmed enteropathy and gastritis by way of scope. She's taking DHIS by Zymogen and started an elimination diet. Okay, so the first thing you want to look for in something like this is the extremes. Okay, so we see some poor alpha diversity. So that's again the quantity of bacteria species, and we see here that red dot is not in the center. It's off the grid here. So that's showing you bad compositional or dissimilarities. And here, we see a score. It's not horrible, but we do see we're dealing with some pathogens, Clostridia and Campylobacter. We look at that 30,000 foot, and wow, look at this. You've got a Firmicutes Bacteroides ratio off the chart. So remember, you're supposed to have more Bacteroides, more red than blue, and man, you got all blue. Okay, so that's a big problem. And that we show here, she has a very low estrobolome, which means the estrogen is being recirculated instead of being excreted. So that's a problem, especially for autoimmune disease. Okay, this just shows the chart just to remind you, the whole when estrogen is being reabsorbed, that it causes havoc. It's supposed to be excreted. All right, now what you want to look for here is just in the keystone strains, look at the not detected, not detected, not detected. So those are problems. Not detected, not detected. All right, so the findings are the dysbiosis defined as loss of beneficial organisms, excessive growth of potential harmful organisms, loss of overall microbial diversity, has excessive Firmicutes coupled with low beneficial microbes that aggravate the autoimmune symptoms, and prone for estrogen dominance, which aggravate the autoimmune systems. So she definitely needs, hormones checked, but she needs her gut bugs fixed. You do that by adding a variety of plant fibers, resistant starches, anti-inflammatory fats, gluten-free, dairy-free, a moderate protein intake, organic preferred. Support the liver and the bile with the artichoke leaf extract, and I know of a great product that has that. Omega-3 supplement, rich in EPA, I know of a great product with that. Turmeric and curcumin reconditions the gut with Megaspore and HU58. Add IgG2000 to bind up those toxins, neutralize the C. diff toxins. Add the Megapre 1 to 2 times per day for extra bacterial diversification. Okay. And here we have case 2 history. We got an 11-year-old female, neurological problems, behavior, sensory issues, sensitivity to light, constipation, limited diet, no supplements. Okay, let's look for the extremes. This is just a chart showing you the gut behavior connection. Here we got microbial metabolites affecting the hippocampus, pain and emotions, cognitive, social behavior issues. Another one showing it's bacterial molecules, LPS and exotoxins affecting the limbic system, causing behavior and emotional problems. So let's look first at the index. We see we got 14 out of 40, bad. Very low alpha diversity, bad. The red dot is off the crosshairs, bad. So not good. Here we see a whole list of pathogens. Got E. coli, Klebsiella, Candida. Got a bad Firmicutes Bacteroides, off the chart. But this is way off. It's out of the off the planet. You got a 1,700 level on this proteobacteria. So what's that look like? Here proteo is supposed to be a little tiny slice here, but here it's it's just taking up the whole pie. It's become the dictator in the society, and we don't want that. So when we realize all of the harm, all the symptoms that lipopolysaccharide affects, especially when it comes to behavior, mood, depression, cognitive decline, memory issues, depression, anxiety, pain. All of these have to do with the dysbiosis in the gut. These diagrams are just another way of looking at it. These are the organisms that are working in her favor, and these are the ones that aren't there and can't help. So that's a problem. All right, these are the keystone strains. Again, look at the extremes: not detected, not detected, not detected, not detected, not detected, not detected. All right, so the findings: you've got to lower the LPS containing bacteria. It's, she's just burning up with inflammation. It's leaking gut into her brain. So if she's got leaky gut, you've got leaky brain. Constipation is most likely because the LPS is completely shut down her bowels. Very poor overall diversity. We know that. You need to eradicate the pathogens, and we use the total gut restoration. We change the environment. We, push them out. And if we need to add some biocidin, we need to add some, then it's fine to do that. But let the organism, let the bacillus spores go as far as they can, because they're smarter than we are, right? Non-existent commensal organisms, we saw that. So the recommendations: high doses of Megaspore, A258, Restoraflor. Remember, this is that overgrowth protocol. Lots of IgG 2000, 12 capsules a day. So, you've got a lot of leeway. Remember, it was 20 grams that has been shown to be safe. You're not even close to that. So that's fine. You need some omega fatty acids, shut down that inflammation. N-acetylcysteine or glutathione. Again, you've got behavior issues. So, that's a great antioxidant to shut all that down. Support nutrient deficiencies. She might have some methylation issues. Might look into that route, because her biochemistry is, she's got to reroute some, pathways. Get that mega prebiotic in there to encourage the keystones, support the liver and elimination phases of detoxification. Make sure that there's no glyphosate. Check the gluten. There's something that's really triggering all that inflammation. So you've got to eliminate any offender. Okay, and this is the last history here. This is a married couple sent in the BiomeFX with a month and a half apart from each other. So the lab didn't know they were married, and they were sent in at different times. Both were born vaginally, both breastfed, both born in cold climates, moved to warm climates, and back to cold climates. Both eat, exercise, lifestyle, even their job is the same. The male exposed to secondhand smoke. This is the only thing that was different. Male exposed to secondhand smoke for 30 years, moldy hay, and contracts squamous cell carcinoma at 60 years old. Went through a whole 7-week chemo radiation, and currently PET scan negative. All right, so here's the comparison. Here is the individual that contracted the cancer, and this is his mate. All right, so what we're pointing out here is how similar these microbiomes are, that there is almost a syncing, just kind of like the way female menses will sync in an environment, and they believe that's through pheromones. Maybe this is a pheromone thing too. Okay, so very similar microbiomes. That's point one. So similar that we've got some quantities that are not just close, but exactly the same number. So that's just kind of, drive that home, that they're very similar. Something to point out on this, here's all the just comparisons. This individual, the mate here, has consumed yogurt almost every day of her entire life. All right, and look here, just to show, yogurt does not seed any probiotic. Just saying. Okay. Again, look, I mean, not similar. I mean, the exact same number in some cases, right? Okay, just interesting. Wait. All right. Now, the difference here is the male who had the cancer has some pretty nasty pathogens, which is indicative of chemotherapy. Okay, so that's to be expected. There's some articles. I put an article in one of the constant contacts that talks about what happens to the microbiome post chemotherapy to help, for those individuals that are helping cancer patients. All right, so the conclusions. This similarity in the microbiomes, we're looking at some kind of syncing environmental adaptation. Maybe, pheromones are involved. Certainly, same lifestyle. We talked about this. Yogurt does not increase lactobacillus. Chemotherapy predictably increases the pathogenic burden. We saw that. Now, questions that will come out is how long is this going to take for repair? What we typically tell people is for every year that you've had a certain condition, you want to add a month of repair. So, we'll just stay tuned on this. We're not really sure. This is the first post-cancer scenario we've had to deal with. Now, this is just off the wall question. Does the mate's microbiome empathize for its traumatized counterpart? Now, I say that just because there was an interesting paragraph in the paper that I gave you the link to. It had all of those other neurotransmitters and hormone charts in it, but this is interesting. The pheromones, of course, are hormones that play an important role in sexual recognition, attraction, and mating behavior, as well as aggression behavior and dominance. Pheromones are also termed ectohormones, chemicals excreted outside the body of one individual and affecting the behavior of another's. So it's showing here that this, in this study, that the most abundant pheromones were, we saw this indicated on the chart, but what it suggests that a role for the microbiota in pheromone regulation. And the study characterizes mate preference between two groups fed different diets. Assortative mating preferences were eliminated with the antibiotics and dependent on specific gut microbes. Together, these findings suggest the mechanism whereby microbiota affect host pheromone levels, which in turn affect mating behavior. So, this is pretty fascinating stuff. Again, who's really in charge here? How much do your gut bugs have to do with your behavior and your mate selection? All right, so we're going to end with that. Please text in your questions. Let me get my phone here to see how we're doing on time. 8:53. Whoa, right on time. Right on time. So I'm going to open it up here for some questions and unmute all. Well, yeah, let's see how noisy it is. Okay, pretty good. So, wow, what'd you all think? Did you learn something new?
Wow, just wow.
Yeah, Ken has a t-shirt idea. It's called the Nerd t-shirt. It's just my firmicutes talking. Okay, so little humor there. Yeah, little nerd.
Oh man, man.
Okay, so
do you have a date when they're going to come out with the next version of the BiomeFX?
Yes, it is the end of July. It was supposed to be the end of this month, but yeah, they postponed it. Because they really, the reason they postponed it is because everybody that has turned in BiomeFX kits in phase 1, they want to give first shot at getting the phase 2 added to it. And we promised everybody that does phase 1 will get phase 2 added at no charge. So they're going back and they're adding all of that data to everyone who's already done the test. So they're going to do that first and then launch it. So that's good.
Yeah, that's perfect because that answers my question about whether to wait to have it done or not. So we can go ahead
Yeah.
and have it done and we can still get that new information.
That's it. Don't wait. Get it now. It's cool information already, and it's just going to get cooler. Don't be shy. Text your information in. Here, I'll hand my phone to Ken if there's some things he can be reviewing.
Hey, Tracy, I have a question.
Okay.
And my question is related to the sample healthy population that everything is being compared to. Is that a diverse population or is that US Americans? And I want to know how that impacts when we're comparing someone that did grow up and live in, another country.
Yep. It is a global population, and yes, it is diverse. So we're talking
Okay.
different races, different ages, different sexes.
Great question.
Mm-hmm.
Hey, Tracy, this is Eva Clark. I just wanted to clarify this, and you may not have this information, but the yogurt impact of the lactobacillus and other species, is this a store-bought yogurt or is this something that people will have a different outcome if they make their yogurt is non-pasteurized and they make it with different strains? So do you know that to be
Yes,
a different outcome?
I don't think there'll be a different outcome because that individual course that we were talking about was me, and I have made my own yogurts, and I have consumed yogurt almost every day of my life. And the reason is because any non-spore organism, probiotic, is going to die in the stomach. It's going to be digested. And even if it had some technology that took it to the intestinal tract, it would not survive. A mature microbiome does not accept new members. And even beside that, even if it did accept new members, the way they make probiotics is in the air, in fermentation vats in the air. And they evolve to lose their facultative anaerobic abilities, and then when you put them in an anaerobic environment, they die. So there's many levels of they die. So
Okay, good, great, thank you. That kind of makes that explanation.
Yeah.
Because
Now,
I have a lot of clients that want to make their own yogurt.
Yeah, and it's not to say yogurt's not good. I mean, all of the fermented products, the kimchis, the sauerkrauts, the yogurts are all good because these organisms produce metabolites in those products that are very healthy for the microbiome. It just doesn't seed the microbiome. Those organisms don't make it there, but all their metabolites do, and that's all good.
Okay, yeah, and plus it's a pre-fermented fiber if they eat different vegetables that are fermented, so
Yes,
that's
and
easier to tolerate for some people.
absolutely. And
okay, great, thank you.
We want to encourage as much diet diversity as possible because we are so narrow in our selections as Americans. So you really want to encourage them to go to international grocery stores, give them lots of tasty recipes to try new things, because people get in a rut and that hurts their microbiome.
Right, and the trend of the ketogenic diet right now is really having an impact, because I have some people that are coming back from having to, they stop having the keto desire and they want to incorporate different vegetables, and they're having a big problem now because they can't break down cellulose. They having the microbial diversity has changed, so now they're having an issue.
Yeah.
So that's good to know
Even-
that you can do the testing and see if they even are a candidate for something like a ketogenic diet or high protein diet.
Yes, and you're going to see that evolution happen in these testing. It's going to become very sophisticated to where you can see what diet is best for a person based on their gut bacteria. I can see a whole new diet plan, evolving from the BiomeFX stool kit. So, that's all going to happen. And as we're talking about food, also pay attention to the oils that people use. We've talked about this in other lectures, that there are certain oils that increase inflammation more than others. And we've got data on that. I have many of those YouTube videos talk about, the coconut oil versus avocado oil versus olive oil versus fish oil. All of those are different as far as the inflammation. So just, FYI, be careful. Coconut oil on a damaged gut is like gasoline on a fire. So you really need to heal the gut before you introduce a saturated oil like coconut. Now, avocado is different. It doesn't have the same response. Coconut does. So be careful, heal the gut up before you introduce coconut oil.
Right. So basically olive oil would be the best option.
Yes, olive oil, as long as you keep it, don't have extreme temperatures, because olive oil, can be damaged and it goes trans. So
Right.
it's good for just, sauteing, low temperatures, the best for, any type of salad dressings, and baking is fine.
Okay, great. Thank you.
You're welcome.
I have another question.
Sure.
When would you use the Restore Flora? What's its purpose?
Super. A couple different ways and reasons you would use Restore Flora. Number one is those individuals, again, that have a difficulty tolerating Megaspore, you can use it as a step up. So you would start them with the 2-spore probiotic and then graduate them to the 5. Another reason is that it has Saccharomyces boulardii in it. So if you don't have time to titrate someone, they end up in your office, they just started a course of antibiotics, and you want to get them some repair right away, then put them on Restore Flora. They're going to tolerate it easily. They're going to have the SAC-B in there, to come against any candida that's going to result from the antibiotic. And they can take it at the same time as they're taking the antibiotic. It's not going to hurt the spores. And this is real important because, the disinformation that you've received all these years about, if, other probiotics actually worked, the fact that it was so confusing when to take them. Well, you're taking antibiotics, so you can't take a probiotic at the same time. So you have to, separate them by 8 hours or a day, or, it was confusing. This, you don't have to worry about. You can take them at the same time. The spores are smart enough. They will stay in the spore stage until the antibiotic passes through, and then their quorum sensing tells them the antibiotic's gone. They can come out, colonize, clean up the damage, and start repairing right on the spot. So good information there.
I have a question.
Yes.
I'm thinking about timing as far as testing. Like if I'm trying to guide how to change a person's diet or what their diet's doing to them, how quickly will we see changes? Like if they've just done a 5-day fast and then they come to me and I get a test, obviously that's going to look better than when they go back to their bad diet. And it's so expensive. I don't want to do a lot of repeat tests. So what would be good timing as far as how long it takes for things to affect as far as diet goes? And
Okay, so there are a couple layers of, now if you're doing a total gut restoration, typically you'll do a BiomeFX test before, and then the very next day, or as soon as after you actually sample the stool, you can start the total gut restoration. And then you would retest, say right at that 3rd month is fine. So typically it takes, 90 days. You got that 3 months, because you're looking for complete cellular turnover. You're changing the ratios. You want to give it enough time to be a representative change. So that 3-month mark is ideal. And now, as far as when a person experiences a change, that will vary. As soon as they start taking Megaspore, if they're not dealing with any intolerances and they have to step down and change, the average person, 80% of the individuals, go through the titration just fine, go through the total gut restoration in 3 months just fine. And it will vary. There will be some people that will say, I didn't really feel any different, but I can eat more foods. I don't go to the bathroom 4 times a day, or, they didn't feel any different, but, listen to what their physiological changes are. But then again, you're going to have other people that are sending you pictures of their stools from the toilet because they're finally having a normal bowel movement, so it really changes. It's different for different people, how they, share their changes. And listen for the people that will say, I haven't felt a digestive change, but I'm in such a good mood. I feel so much more energy. Listen for the behavior type of things too, because it's so interconnected that does relate.
So specifically for fasting, I have a lot of patients that come to me who've been fasting, and they'll come like the day after they fasted and they'll say, well, I feel great today because I've been fasting, but give it 2 weeks, going back to my diet. And so I'm kind of curious, how long do I need to wait to do the test after they fasted to show maybe what's more normal for them when they're in an eating pattern?
Fasting. If fasting is a part of their regular life, you can do the testing anytime. If it's anything that is out of the norm for them, then get a sample of what is the norm for them. Let them, finish their fast, get a week into their regular eating habits, and then sample. Try to get a representative sample of their life. If they normally take supplements, sample when they're taking supplements. Don't sample an extreme situation that may never come again. Try to get a normal. And then when you're talking about intermittent fasting, this is an important point too, because you're looking at what environment do your gut bugs prefer. And something like Akkermansia, it gets excited when you're not eating. So when you fast, you increase that organism. It likes to, gets an advantage, and you want more Akkermansia because it's a fat burning organism. So, the rule of thumb is if you can, a normal fast, when you're sleeping is 8 hours. And when you don't eat after 8 in the evening and then you eat again at 8 in the morning, then that's 12 hours. So that's pretty normal. For someone with a damaged gut, you want to try to increase their fasting time so they have more gut healing. So you want to increase it, from 12 to 14. And then for extreme damaged conditions, you want to try to increase it to 16, so that you're not eating 16 hours and you're just eating within an 8-hour period. So that's a very healthy intermittent fast when you're trying to heal a damaged gut.
Tracy, can I ask a follow-up to that?
You bet.
Does taking other medications like thyroid medication, does that break the fast, or you're just talking about food?
I'm just talking about food.
I have a lot of clients on thyroid meds.
Right. And
Okay,
you know,
so somebody could take their thyroid medicine in a smaller window and then still wait to eat.
yes, yes. And natural thyroid medications generally are taken throughout the day, you know, so that it better mimics a functioning thyroid. So it's not uncommon. That's the way I don't have a thyroid. So I take, you know, a dose at 6, at 10, and at 2, just to give me more stability through the day. So yeah, you don't want to interrupt those type of regimens.
Okay, great. And can I also ask, if somebody has a pretty serious parasitic infection, I hear what you're saying, that it's all about supporting the microbiome rather than killing it. I'm just wondering, do you have, are there studies that show the evidence that they actually are being crowded out, and that then parasitic infection clears up, or does it just go down so it's not bothering the person anymore? I'm just curious, like, does it completely clear out, or is there ever a case that you would recommend doing an antibiotic protocol for somebody and then doing the total gut restoration program?
Yeah. Before BiomeFX, I would say everything that we have was anecdotal and case studies. We probably have, you know, 10,000 individuals globally that are using the product. So really good results using it with parasites. But there are cases that are extreme that did require antibiotics. Some were already on a course of antibiotics when they arrived at the practitioners, and it was really a matter after that of just cleanup. Now with the test, we're going to see more specifically what's going on behind the scenes, and it's really just a matter of time and, you know, staying, you know, close as far as, you know, as we're doing more of these lectures and getting more practitioners experienced with the test, reporting their findings, we're going to nail down a lot of what's been happening and be able to explain why and how.
Gotcha. And does the total gut restoration program, does that, you know, if somebody has done a couple rounds of antibiotics, does one round of the total gut restoration program, so 3 months, does that tend to bring it back into balance, or do you need to do sometimes more than one round to counter all the antibiotic use?
Yeah, it's still really individual, because it's more than just they were on a course of antibiotics. You know, were they vaginally born? Were they breastfed?
Yeah.
What was their underlying developmental microbiome before they had the antibiotics? You know, there's a good study out there that shows, depending on their microbiome, one course of antibiotics might be cleared up in 10 days, or it might damage the person for a year, or it might cause permanent damage, depending on, you know, how vulnerable that person was.
So permanent damage that even the total gut restoration program couldn't necessarily
No,
turn around.
That's just saying in general. You know, I think we haven't had anybody do the total gut restoration that hasn't benefited. So it's really a matter what's the history, what's the genetics, and, you know, how compliant are they. You're going to get some variability, but you're always going to get benefit. We haven't had anyone not.
Gotcha. And I just want to make sure that yeah, go ahead.
I was going to say, I think by permanent damage, what they mean is permanent changes in the microbiome, that not damage to the gut necessarily, but their microbiome has been permanently damaged to the point that it can't restore itself back to what it was before they took the antibiotics.
Yes, there are some signs of that. I think you can, if you, for instance, received just a very small amount of your Akkermansia and Faecalibacterium, and if you pounded it, with a heavy dose of antibiotics, you could permanently damage those and not be able to bring them back. Now, it's not to say that the total gut restoration
Okay.
wouldn't be able to bring back other keystone strains, so you still have some friendlies that are going to buffer, your resistome and going to, keep you, giving you some benefit where otherwise you would be completely vulnerable.
Gotcha. And I understand that for SIBO that the Megaspore biotic can be really helpful, but did I hear that even the prebiotic is okay for SIBO?
Yes, a lot of our SIBO practitioners will burn on the prebiotic very slowly, but the whole idea is that, even if you have SIBO, you still have keystone strains that need to be fed. And those oligosaccharides are very specific.
Yeah.
They're not going to feed the pathogens.
Gotcha. And because motility is such an issue with SIBO, would you recommend for a SIBO protocol also doing, the mega mucosal?
Yes, the total gut restoration program and the addition of the MegaGuard, just because most SIBO issues
Yeah, that's what
also,
I meant, the MegaGuard.
Yeah, also have that bile issue, and as practitioners, whatever helps
Right.
you need, like magnesium, whatever, to keep the kinetics moving, every practitioner has some favorites that they will add in there too.
Excellent. Thank you.
You're welcome.
I was going to say, when we were talking about you having some folks go into it not noticing very much difference unless you really talk to them. That's why I think it's really important to do a really thorough symptom questionnaire or something like that before they start, and then again one maybe
Absolutely.
midway and at the end, so that they can see their progress, and you can too.
Absolutely, that's critical. And anyone who's dealt with the general public, you have to train them how to help themselves. And it's just human nature. When they start feeling better, they forgot the symptoms they came in with to begin with. Well, this has been a blast, and I really appreciate everyone that joined us. I hope that everyone will schedule some follow-up Zoom calls so we can really continue these relationships and enhance your understanding, answer all of your questions, get you really comfortable with us and with the company and with the products. Just want to extend that invitation. So please take us up on that and join us for the, the Nerd Hour every Thursday, 8 Central Time. So you can, keep the relationship going with some of these other practitioners. Iron sharpens iron. And I like challenging questions. And the group is getting real skilled, as well as breaking down different difficult gut conditions. And it's just always good to hear from someone else's perspective. So I hope this was beneficial. Keep those questions coming. If you wanted to place an order, you need help, I'm happy to walk through, different scenarios you might be dealing with, so you get the right combinations and get the best prices. So, okay, super. Yeah, you can email those in, or you can give us a call if you have some questions about it. So how we
Are
doing
you going
on-
to talk about the discount?
Yeah, so the discount is 10%, and it's going to be after you've mixed and matched to get the volume discounts. So the first hour, if you listened to that, will give you a discount on Megaspore, Megapre, and Megamucosa. And then the second hour will give you the 10% on the BiomeFX registrations. And you can use these codes for multiple orders. So if you've got multiple stool kits you're sending in, you know, line them up. Let's use the code on all of them. You've got a week. It's best to try to get it the orders in as soon as possible. Next week's kind of hairy. We've got national training. I can help you in the afternoons, but you do have a week to look at your inventory, think about your, you know, customers, you know, who will benefit from what, and rack up the discount. Yeah, and if I said it wrong, it's 10% on top of your volume discounts. So you can mix and match. So when you mix and match, say 3 Megaspore, 2 Megapre, and 1 Megamucosa, that's 6. So that triggers a 5% discount with your, and then it'll add 10. But if you get, you know, 4 times those, you get the 12 bottles, which triggers a 15% discount, and then the code will give you 10 on top of that. So 25% is really handy. Rack up some savings there. So let me help you work those through if you need it. Otherwise, email those in. I'll process them with the code. And yeah, stay in touch. This has been great. I'd like to do more in the future. This is our first one. And I hope you got a lot out of it. Give me some feedback. If we can do a better job, we will. So, can I see my phone? Second.
How do we join the Thursday coffee hour? Where's that at?
It's the same Zoom link every week, and I will email that to you, and you just show up. And it's come as you are when you want. And we, like I said, we have a nice group that are loyal. There's usually, you know, 6 or 7 of us, and if I don't have a formal topic to cover or a new product that's coming out, then we just open it up to strategizing protocols and answering questions. So join us. The more the merrier. And it's the
It is
same.
And how much is the
extremely helpful.
BiomeFX?
Oh,
Bell here.
sorry about that. Go ahead.
Bell here.
How much is the BiomeFX?
The BiomeFX to register is $299. So if you use the discount for listening today, you get $29 off of that. Yeah, so order the
Great.
kits.
Thank you so much, Tracy.
You're very welcome. And know that the Zoom link you used for today is the same Zoom link for Thursdays. So that's how you join. And Kevin, Dr. Kevin, you were going to say something?
Yes, it is extremely helpful those Thursday mornings.
Super,
Highly would advise people to attend.
super. Yeah, we've got a good group going, and yeah, it's great to hear from other people.
We have a doctor all the way from Africa.
Yeah, and Dr. Sam sometimes joins us from Africa. So we have a global group.
And will there be a recording of this talk if there's something we want to go back and review?
Yes, yes. In fact, that's a good reminder for me to stop the recording so it's not so long.